Now, as I said a few times, the protein structure determines its function. So let's talk about protein function. It too, the protein functions have also been grouped into several classes, in this case 3. The first class of proteins, of protein function is the reversible binding class. So an interaction between a protein and a ligand of some kind. Represented schematically as a colorful blob. You can see that this is the protein, and it has an area which will interact with the ligand, which is blue. It will bind the ligand, and it will be also capable of letting the ligand go. And this has to be a reversible process, because so that you don't have to lose a whole protein just because you bound the ligand to it, and it's not functional afterwards. Now, elaborating on this whole binding function is the catalysis function. So all the enzymes that you have, you guys are going to cover in biochemistry. Now, an enzyme, again here in red, is capable of binding the blue thing. In this case, it's called a substrate. But unlike just a reversible binding, the enzyme is also capable of changing the substrate from blue to orange. And most enzymes, unless ATP has been used or other tricks have been employed, are also reversible. They are just as happy binding orange and converting it back into blue. And then finally, another functional class of proteins is the proteins capable of motion or motor activity, so kinesins, helicases, and other proteins. And if you guys want to see a cute video of a kinesin motion, so that, a movie is worth 1,000 words, you can go to this YouTube video and play it. I can't play it due to copyright. And what I would like to point out, however, is that many proteins are able not just to perform one type of function, but they are able to do multiple, such as the RNA polymerase that can bind DNA, that's reversible binding, catalyze RNA synthesis, that's catalysis, and it can move along the DNA in one direction. These proteins that have multiple functions usually are multi-subunit proteins that take where each subunit can fulfill one of its functions. So let's talk about the reversible binding function of proteins. This is your typical globular protein that is holding a ligand. It's called a complex between a protein and ligand. So the ligand is anything that can bind to a protein, and the word ligand implies that the connection is temporary, and though sometimes it's not the case. In this case, the ligand is ATP. You can see the adenosine part of the ATP and then the 3 phosphates. The binding of the ligand to the protein occurs in the location called the binding site. So that's the area of the protein that can really well interact with ligand, specifically better than water. So it's usually will be complementary in size, shape, charge, or the hydrophobicity or hydrophilic profile of the side chains present inside the protein in that area. And then finally, how is this binding done? Very frequently, there will be at least a slight conformational shift inside the binding site once the ligand has entered. Again, to prevent interaction of the ligand with the water and directing it away, just holding it there. But the induced fit has to be definitely flexible, because it's very important for the proteins to not only be able to bind the ligand under one condition, but also to let go under another condition. And one way to look at these binding interactions between a protein and a ligand is using this dissociation constant. So in this case, equations have been derived that would allow us to define the different dissociation or association abilities between a protein and a ligand to see which proteins bind tighter and which proteins not so. In this case, the equation is really simple, and you can see that the Kd, the dissociation constant of the interaction, is the ratio of K off to K on, or the concentrations of the protein and ligand separately versus the protein and ligand together. And you can calculate both Kd and Ka, but most of the time Kd is calculated since it's simply easier to calculate. You can make a bunch of your protein and your ligand, saturate your protein with your ligand so you know that all the binding sites have been filled, and then measure how much of the ligand is left over in your solution that hasn't been saturated. And from that, since you know the concentration of the ligand and you knew the concentration of the protein, you can start to calculate the Kd. So here, the idea is displayed in a graphical way how the actual experiment was performed. So you can see the concentration of the ligand plotted on the X and the fraction of the ligand bound to the protein on the Y. And once saturation has been achieved and you know that your graph is plateauing, you can estimate what's the theta for the fully saturated protein, and then half of that is the Kd of the protein. So half of the ligands have been bound up by the protein. And dissociation constant will differ. What's important to understand about the dissociation constant, that different proteins will have different ones, and the lower the dissociation constant is, the tighter the protein binds the ligand. So in this case, the graph for red shows a very, very tight binding with a Kd of 1 nanomolar. So only at 1 nanomolar concentration have most of the protein sites have been occupied by the ligand. Half of the protein sites have been occupied by the ligand, versus the green graph where the interaction between the protein and the ligand is fairly transient and the complex dissociates easily. So here, this graph from this table from your book shows the average Kds for different protein-ligand interactions. So biotin and avidin have a typically strong interaction with Kds at 10 to the minus 15 molar. While the more typical receptor ligand lay in this range. You can see that antibody interact a little bit stronger than enzymes and substrates, or several fold stronger than enzymes and substrates as antibody-antigen. The reason for an antibody-antigen interaction is to not let go, while the reason for an enzyme and substrate interaction is to convert the substrate and then use another substrate molecule to continue catalyzing the reaction. One important thing that I would like you guys to take away from it is that you can have specific and non-specific interaction between proteins and ligands, as shown here. So lactose repressor is a protein that binds DNA. It binds a particular sequence of DNA, but it can also interact with DNA non-specifically. So you can see that there's a many-fold difference in the interaction between the non-specific binding of the lactose repressor to DNA versus the specific binding of the lactose repressor to the very particular DNA sequence that it prefers to recognize. However, do appreciate that all proteins can bind non-specifically to other proteins. It's just very, very high Kds. All right. So one example of reversible binding that's very topical for DNA replication and other subjects of molecular biology is the ability of proteins to interact with DNA. Because in order for proteins to manipulate DNA, whether replicate it or make RNA from it, proteins have to be able to find it. They have to be able to bind it. So it's a protein and ligand interaction. And there's two big types of DNA binding proteins. There's a non-specific DNA binding protein, such as histones, which interact with the negatively charged phosphates in the phosphodiester bonds of the backbone of the DNA, which are the same, just no matter which nucleotides have been incorporated inside. And there's sequence-specific DNA binding interactions between the proteins in the DNA. An example of that are transcription factors, which are proteins that help to recruit RNA polymerase to the area that needs to be converted into mRNA. These proteins, instead of utilizing the backbone of the DNA, have to recognize the actual nucleotides, have to be able to form hydrogen bonds between the actual nucleotides of the DNA sequence to make it specific. Usually it's done via hydrogen bonding, though there's some also hydrophobic interactions, and they occur usually at the major groove of the DNA helix. So you can see here a picture from your book of non-specific DNA binding protein interaction. The molecule in the middle here is the DNA, really only one part of the one strand of the double-stranded DNA helix. And you can see how the arginine, positively charged amino acid arginine and positively charged amino acid lysine are interacted with the negatively charged phosphates of the hydrogen backbone, of the phosphodiester bonds of the DNA backbone. Now, how is the sequence-specific DNA binding achieved? So here is a picture of a transcription factor interacting with the DNA molecule, and you can see that the DNA molecule is sitting inside the major groove of the DNA. So see how this space is a bit wider than this space. This is the minor groove of the DNA helix. This is the major groove. And because of the orientation of the bases inside the major groove, some parts of the nucleotides are exposed to either the water or to be able to interact with a protein. So you see this is the major groove area. The orientation will expose this part of the base pairs. And you can see that this nitrogen and this hydrogen will be able to form hydrogen bonds with a protein as well. In the AT base pairs, this nitrogen and the adenosine and this hydrogen and adenosine will also be able to base pair. So one example of such base pairing is interaction of arginine, which is a positively charged amino acid, with the negatively charged nitrogen and hydrogens of the guanine and the cytosine, as well as the interaction of glutamine with the AT base pair here. All right, let's get back to the protein function of, specifically the sequence-specific DNA binding. Sorry, the computer crashed. So you can see here that the amino acid arginine is capable of making hydrogen bonds with the GC base pair, as well as the glutamine will be able to base pair to an AT. So these amino acids will be found inside the binding site of this particular DNA binding protein if it needs to interact with G's or G's or A's. And that's how, if you put a bunch of these binding sites in a row, you will be able to recognize very, very specific sequences inside DNA. And that's how transcription factors make their decisions. Now, let's move on to the next type of protein function, which is catalysis. There's another typical globular protein, monomer, and in this case it's an enzyme, and it has, just like a protein that binds, has very particular architectural features. So in the case of an enzyme, in case of catalytic function of proteins, and those proteins are usually called enzymes, the molecule to bind is called a substrate. Now, it's a molecule that will be undergoing a chemical transformation inside the enzyme. So the ligand doesn't change when it binds to a protein. However, a substrate does. And just like the ligand was binding to a binding site, the substrate will be binding inside an active site, which is an area of the enzyme that has particular chemical conditions that will be perfect for, or at least optimized for coaxing the substrate into transforming. And how do enzymes accomplish this? Well, first of all, by providing an active site that stabilizes the transitional state of the substrate to product. And they do it by using a particular induced fit, just like ligand binding was, or the cooperativity between the different subunits of a multiproteins complex that allows for particular shifting of the substrate to stabilize several transitional states, or by using more chemically active prosthetic groups if the amino acid side chains cannot do the job. So a lot of the enzymes incorporate vitamins or various metal ions in order to help them catalyze reactions. So this is an example of one particular active site, again using the RNA polymerase. So in this case, the substrate are the nucleotides, and the product is the RNA being produced. And the chemical reaction is a synthesis that the RNA polymerase enzyme catalyzes, is a formation of the phosphodiester bonds and the release of conversion of the ATP, CTP, GTP, and UTP into a polynucleotide stretch, aka RNA. And then you can see that the way the active side of the RNA polymerase works is it has a few glutamates in a particular orientation and uses the acid-base chemistry to allow for electron flow, electron transfer from the base down to the acid. And as part of this electron transfer, the phosphodiester bond between the first phosphate of this molecule and this molecule will be catalyzed to form the RNA. Now, let's think about catalysis a little bit more in terms of in chemical terms. So basically, if there were no enzyme to catalyze a reaction, and in this case the reaction is we would like to break a metal stick, somehow we would still have to force this transition state from the substrate to the product. Now, in the chemistry lab or at home, you could take the metal stick. If you have the Bunsen burner, you can heat it right there, which will soften the metal and allow you to break the bond between inside the metal. Or, since cells don't have a Bunsen burner, you can use an enzyme. Now, just like the Bunsen burner is not used up in the process of breaking, though, I mean, I guess gas is used up. The enzyme itself is not changed during the catalysis. That's the principle of catalysis. A catalyst stabilizes the transition state of a substrate to product conversion, allowing for less activation energy to be used in order to create the product. So in this case, the book depicted the enzyme as providing these magnets, these little red things, that will force the transition state onto the substrate, bending it, which will then provide enough, will be sufficient for the metal stick to break. So in chemical terms, enzymes increase the rate of reaction by lowering the activation energy. In the biological terms, what enzymes do, they make the high activation energy activation energy reactions possible under biological conditions because we exist at a very low range of temperatures, and enzymes can only exist at a very low range of temperatures. simply because they themselves are held together by hydrogen bonds, which are very weak bonds, and any excessive heat will break them first. So it's the shape and the chemical environment that the active site of the enzyme creates that will allow the stabilization of this transition state that you can see here. So let's look a little bit more at the enzyme catalysis using this. What I want to point out here, and what the book I think really brings down very nicely, is the fact that the enzyme does not stabilize the substrate. This interaction here is your typical protein-ligand interaction, where the protein does not change the ligand. It provides a perfect fit for the ligand. The stick really likes to fit inside this magnet-lined enzyme, but it's incapable of changing it in any way. The enzyme, on the other hand, is not the perfect fit for the substrate. The substrate will have to conform to fit into it because it wants to fit in it because there's magnets. But to fit into the magnet line side, it will have to assume the transition state, which will then make it much easier for the stick to break, because it has already been bent in order to fit into the enzyme active site. And this diagram, which just graphs out the delta G's and the changes in delta G's that the enzyme is able to achieve. So this sort of reaction without the enzyme would be this black line where a really large activation energy is required, so the Bunsen burner, in order to have the stick break. However, because the transition state, ES in this case, has been fit into the enzyme, much less energy is required for the product to be produced. So, as said in the previous diagram, enzymes increase the rate of reaction by lowering the activation energy. So, now that we have talked about the very basics of how an enzyme fulfills its job, it stabilizes the transition state between substrate and product, allowing for more favorable conversion of substrate to product or product to substrate by lowering the activation energy necessary for acquiring the transition state. The other thing I wanted to talk about is enzyme kinetics, which is a discipline of biology that determines that calculates the rates of enzymatic reaction and tries to understand how experimental parameters affect an enzymatic reaction. It's a very old discipline, and it's still very important in understanding of how enzymes work. And most enzyme catalyzed reactions can be described by the Michaelis-Menten equation, which was derived in the 19th century by Leonard Michaelis and Maud Menten. And here's the equation, which states the initial velocity equals the Vmax of substrate over the Km plus substrate concentration. In the way Kms and Vmaxes and V0s are experimentally calculated, and they are experimentally calculated, that's the whole point, is to take a defined concentration of the substrate and a very small amount of the catalyzing enzyme, because enzyme shouldn't be changing, and then allow the reaction to proceed, and then measure the appearance of the product over time. So many time points are collected, and a curve is fitted against those time points. It's usually, and the important thing to recognize is that at the very early beginning of the reaction, the curve is not a linear line can be fitted to the curve to determine the initial velocity, which is this point of the reaction, how fast the reaction was going at the full 2 micromolar concentration of the substrate. So then experimentally, all the V0s are calculated for different substrate concentrations, and Here you have the V0 for 10 micromolar, 5 micromolar, 2 micromolar concentration of the substrate. Here, here, and here, so you will get a number. And then this number is then plotted again to get this particular curve. And you can see that the very, very high substrate concentrations, V0, basically plateaus along this imaginary line, which is Vmax. It will never reach it, but you can, knowing how the trend of the curve is going, you can estimate Vmax. Now that you know Vmax, which is the maximum velocity of the reaction, half of it, which is, so you see, V0 plotted here, here's Vmax, half of the Vmax. You extend the line of the curve, and then you get a number of the substrate at, what is the concentration of the substrate when the V0 is half of the maximum velocity, which is the Km, or the Michaelis constant. Now, the Michaelis constant has been calculated for many, many enzymes. Every time somebody discovers a new one, they will have to calculate the Km. And an enzyme's Km describes how the substrate concentration, again, at what substrate concentration half of the enzyme's active sites are occupied by the substrate. In a case of a high Km means that a lot of the substrate must be present in order to saturate the enzyme, so the enzyme's affinity for the substrate is low. So if we go back to the previous picture, you can see that if Km is high, that means this curve is low, and that means that the enzyme is not very, the enzyme has a very low affinity for the substrate. And now a low Km, in, for example, that of lysozyme, means that only a small amount of substrate is needed to saturate the enzyme. So an enzyme has a very high affinity for the substrate. And you can see in this particular table that you don't need to memorize, I just here as an example, that one enzyme has a bunch of binding sites, for its different substrates, because it's catalyzing complicated reaction of conversion of glutamate to alpha-ketoglutarate. And you can see that the product, in this case alpha-ketoglutarate, has a high Km, so enzyme has a low affinity for it, while glutamate has a much lower Km, so the glutamate dehydrogenase has a high affinity for glutamate. As a result, the reaction is likely to go glutamate to alpha-ketoglutarate, even though both directions technically are possible due to chemical equilibrium. Right, so then the final protein function I want to briefly talk about is the motor activity of some proteins. Examples are, again, kinesins, and in this case it's a helicase. So DNA helicase unwinds DNA, allowing for RNA, either for DNA to be replicated or RNA to be transcribed, because you need to open the helix. And motor proteins, because of their complicated behavior, they actually do turn and move and shimmy and click and kick and do all kinds of cool stuff. Motor proteins combine in their function several aspects of previous functions, so they can have to both bind a ligand, in this case, is stuff they're going to move on, and be able to catalyze. In this case, most of motor proteins catalyze ATP in order to get the energy for the conformational change that's required for motion. You have to walk somehow. So again, yes, so the motor proteins will bind a certain surface, a ligand, over which it will move. Movement will be achieved by a conformational change in the subunits of this helicase, or the conversion from an alpha helix to an unstructured sheet and a kinesin, allowing for this for the step of the kinesin to happen. And then conformational change is powered by the conversion of ATP to ADP, so ATP hydrolysis. So in this, looking from that perspective, a motor protein is functioning as an enzyme because it catalyzes the conversion of ATP to ADP. So in this case, this diagram shows how this helicase, which has 6 subunits, rotates along the DNA, forcing the base pairs apart and causing the DNA to unwind, basically melting the DNA double helix, just like high temperature would do so. It lowers the activation energy for the melting of the DNA double helix. And you can see how the green subunit first rotates clockwise in this picture to this position, then to this position, and then back to its old position. For every third of the circle rotation, ATP is hydrolyzed into the subunits and converted to ADP, which allows for conformation change in the subunit and allows for this rotation, because in order to fit the enzyme perfectly to the DNA. And this link over here from YouTube is just a video of how a helicase works. It explains it a little bit to greater detail. But as long as you guys remember what motor activity entails, that it entails binding, it entails a conformational change, and enzymatic activity, aka ATP hydrolysis, I'm good. So this is a summary slide for you guys to review before the quizzes. I think it's kind of extraneous for me to read it for you. Just read it yourself. Again, the PDF for this lecture is in the Live Courses file section that you can download